Hello, welcome to the instruction video on enzymatic activity of laccases in soil. Laccases are extracellular enzymes belonging to the group of oxidoreductases. Specifically, laccases are multi-copper oxidases and blue multi-copper oxidases. 4 copper molecules present in the active sites of laccases participate in oxygen declination and water production. Laccase is considered an eco-friendly enzyme and is also known as a blue enzyme. Laccase could aid in degradation of hazardous organic compounds, for instance phenols and chlorophenols. Laccase is widely distributed in plants and fungi, effective in degrading phenol and lignin. The activity and stability of laccase produced by white-throat fungi, oyster mushroom, can be enhanced by addition of copper, cadmium, silver, mercury, and lead to prevent the enzyme activity. Along with the other lignin-degrading enzymes, laccases can catalyze the degradation of polycyclic aromatic hydrocarbons. And how to measure the activity of laccases in soil? First of all, you need to prepare chemicals which will be used during extraction and measurement. There are solutions of phosphate and citrate phosphate buffers, and the ABTS solution for color reaction. The phosphate buffer will be used for the extraction. As the laccases are extracellular enzymes, soil samples can be analyzed in a freeze-dried form. Soil should be ground and sieved through a 2 mm sieve prior to analysis. The common mass of sample for the analysis is 200 mg. Plastic Erlenmeyer flasks are suitable for the extraction. When the sample is weighed, 20 ml of the 50 mmol phosphate buffer is added, and the mixture is briefly homogenized using the high-speed batch homogenizer. The homogenizer must be washed using demineralized water after each sample to avoid cross-contamination. Right after homogenization, sample suspensions must be filtered through filter paper in order to remove solid particles and obtain a solution where enzymes are dissolved. The step of desalination follows the filtration. To do so, desalting columns are used. The principle of desalting is based on size exclusion chromatography, so the molecules are separated on the basis of differences in size. You can see the working procedure on the screen. Pipette is set to the volume of 3.5 ml, and demineralized water is introduced to the columns. This water serves to flush the columns. When water stops dripping, the sample can be introduced. 2.5 ml of the sample solution is pipetted into the column. The leachate is still not collected because flushing water is pushed out from the column by the sample solution. When columns stop dripping, test tubes for sample collection are placed under the columns and 3.5 ml of water is added to the columns to push out the desalinated sample solution into the test tubes. Finally, 3.5 milliliters of water is pipetted to the columns in order to wash the columns. Here you can see the design of microplate. For laccase activity, 24 samples can be analyzed on one microplate. It is important to measure each sample in at least 4 replicates. Each sample is represented by one color. Start with the transferring of the sample extracts into clean and marked reagent reservoirs and pipette into the microplate according to the scheme. Change the pipette tips between the samples. Next step is to distribute the substrate ABTS and citrate phosphate buffer solutions into the microplate. Start with 150 microliters of citrate phosphate buffer into all wells, and then 50 microliters of ABTS is pipetted again into all wells. Right after pipetting the last solution, microplate must be placed into the reader to start the measurement. The principle of the measurement is based on color change of the solution caused by the oxidation of ABTS to its corresponding cation radical, giving the color change from slightly yellow to intensely turquoise. The intensity of resulting color is determined at 420 nanometer wavelength. The measurement is done in 7 cycles after 2 minutes each. It takes around 12 minutes to analyze the whole microplate. Now is the time to answer displayed questions. In case you miss an answer, we recommend watching the video again.